Photo shows a variety of cheeses, fruits, and breads served on a tray. Figure 3.1 foods such as bread, fruit, and cheese are rich sources of biological macromolecules. Credit: Modification of work by Bengt Nyman. Chapter outline: 3.1 Synthesis of biological macromolecules. 3.2 Carbohydrates. 3.3 Lipids. 3.4 Proteins. 3.5 Nucleic acids. Food provides the body with the nutrients it needs to survive. Many of these critical nutrients are biological macromolecules, or large molecules, necessary for life. Different smaller organic molecule, monomer, combinations build these macromolecules polymers. What specific biological macromolecules do living things require? How do these molecules form? What functions do they serve? We explore these questions in this chapter. Learning objectives. By the end of this section, you will be able to do the following: understand macromolecule synthesis, explain dehydration, or condensation, and hydrolysis reactions. As you've learned, biological macromolecules are large molecules, necessary for life, that are built from smaller organic molecules. There are 4 major biological macromolecule classes: carbohydrates, lipids, proteins, and nucleic acids. Each is an important cell component and performs a wide array of functions. Combined, these molecules make up the majority of a cell's dry mass. Recall that water makes up the majority of its complete mass. Biological macromolecules are organic, meaning they contain carbon and are bound to hydrogen, and may contain oxygen, nitrogen, and additional minor elements. Dehydration synthesis. Most macromolecules are made from single subunits, or building blocks, called monomers. The monomers combine with each other using covalent bonds to form larger molecules known as polymers. In doing so, monomers release water molecules as byproducts. This type of reaction is dehydration synthesis, which means to put together while losing water. Shown is the reaction of 2 glucose monomers to form maltose. When maltose is formed, a water molecule is released. The components of the linkage are uppercase O uppercase H from one glucose molecule combining with one uppercase H from the second glucose molecule. Figure 3.2 In the dehydration synthesis reaction above, 2 glucose molecules link to form the disaccharide maltose. In the process, it forms a water molecule. In a dehydration synthesis reaction, figure 3.2, the hydrogen of one monomer combines with the hydroxyl group of another monomer, releasing a water molecule. At the same time, the monomers share electrons and form covalent bonds. As additional monomers join, this chain of repeating monomers forms a polymer. Different monomer types can combine in many configurations, giving rise to a diverse group of macromolecules. Even one kind of monomer can combine in a variety of ways to form several different polymers. For example, glucose monomers are the constituents of starch, glycogen, and cellulose. Hydrolysis. Polymers break down into monomers during hydrolysis. A chemical reaction occurs when inserting a water molecule across the bond. Breaking a covalent bond with this water molecule in the compound achieves this, figure 3.3. During these reactions, the polymer breaks into 2 components. One part gains a hydrogen atom H+, and the other gains a hydroxyl molecule, OH, from a split water molecule. Shown is the breakdown of maltose to form 2 glucose monomers. Water is a reactant. The water molecule, uppercase H subscript 2 baseline uppercase O, breaks apart, with upper O upper H obtained by one of the glucose molecules and upper H obtained by the second glucose molecule. Figure 3.3 in the hydrolysis reaction here, the disaccharide maltose breaks down to form 2 glucose monomers by adding a water molecule. Note that this reaction is the reverse of the synthesis reaction in figure 3.2. Dehydration and hydrolysis reactions are catalyzed or sped up by specific enzymes. Dehydration reactions involve the formation of new bonds, requiring energy, while hydrolysis reactions break bonds and release energy. These reactions are similar for most macromolecules, but each monomer and polymer reaction is specific for its class. For example, catalytic enzymes in the digestive system hydrolyze or break down the food we ingest into smaller molecules. This allows cells in our body to easily absorb nutrients in the intestine. A specific enzyme breaks down each macromolecule. For instance, Amylase, sucrase, lactase, or maltase break down carbohydrates. Enzymes called proteases, such as pepsin and peptidase, and hydrochloric acid break down proteins. Lipases break down lipids. These broken down macromolecules provide energy for cellular activities. Link to learning. Visit this site to see visual representations of dehydration synthesis and hydrolysis. Learning objectives. By the end of this section, you will be able to do the following. Discuss the role of carbohydrates in cells and in the extracellular materials of animals and plants. Explain carbohydrate classifications. List common monosaccharides, disaccharides, and polysaccharides. Most people are familiar with carbohydrates, one type of macromolecule, especially when it comes to what we eat. To lose weight, some individuals adhere to low-carb diets. Athletes, in contrast, often carb load before important competitions to ensure that they have enough energy to compete at a high level. Carbohydrates are, in fact, an essential part of our diet. Grains, fruits, and vegetables are all natural carbohydrate sources that provide energy to the body, particularly through glucose, a simple sugar that is a component of starch and an ingredient in many staple foods. Carbohydrates also have other important functions in humans, animals, and plants. Molecular structures. The stoichiometric formula, CH2O(N), where N is the number of carbons in the molecule, represents carbohydrates. In other words, the ratio of carbon to hydrogen to oxygen is 1:1 in carbohydrate molecules. This formula also explains the origin of the term carbohydrate. The components are carbon, carbo, and the components of water, hence, hydrate. Scientists classify carbohydrates into three subtypes: monosaccharides, disaccharides, and polysaccharides. Monosaccharides. Monosaccharides, mono equals one, saccharide equals sweet, are simple sugars, the most common of which is glucose. In monosaccharides, the number of carbons usually ranges from 3 to 7. Most monosaccharide names end with the suffix -ose. If the sugar has an aldehyde group, the functional group with the structure RCHO, it is an aldose, and if it has a ketone group, the functional group with the structure RC equals OR, it is a ketose. Depending on the number of carbons in the sugar, they can be trioses, 3 carbons, pentoses, 5 carbons, and or hexoses, 6 carbons. Figure 3.4 illustrates monosaccharides. The molecular structures of glyceraldehyde, an aldose, and dihydroxyacetone, a ketose, are shown. Both sugars have a 3-carbon backbone. Glyceraldehyde has a carbonyl group, C double bonded to O at one end of the carbon chain with hydroxyl OH groups attached to the other carbons. Dihydroxyacetone has a carbonyl group in the middle of the chain and alcohol groups at each end. The molecular structures of linear forms of ribose, a pentose, and glucose, a hexose, are also shown. Both ribose and glucose are aldoses with a carbonyl group at the end of chain and hydroxyl groups attached to the other carbons. Figure 3. 4 scientists classify monosaccharides based on the position of their carbonyl group and the number of carbons in the backbone. Aldoses have a carbonyl group, indicated in green, at the end of the carbon chain, and ketoses have a carbonyl group in the middle of the carbon chain. Trioses, pentoses, and hexoses have 3, 5, and 6 carbon backbones, respectively. The chemical formula for glucose is C6H12O6. In humans, glucose is an important source of energy. During cellular respiration, energy releases from glucose, and that energy helps make adenosine triphosphate, ATP. Plants synthesize glucose using carbon dioxide and water, and glucose in turn provides energy requirements for the plant. Humans and other animals that feed on plants often obtain glucose from catabolized cell breakdown of larger molecules, starch. Galactose, part of lactose, or milk sugar, and fructose, found in sucrose, in fruit, are other common monosaccharides. Although glucose, galactose, and fructose all have the same chemical formula, C6H12O6, they differ structurally and chemically, and are isomers because of the different arrangement of functional groups around the asymmetric carbon. All these monosaccharides have more than one asymmetric carbon, figure 3.5. Visual connection. The molecular structures of the linear forms of glucose, galactose, and fructose are shown. Glucose and galactose are both aldoses with a carbonyl group, carbon double bonded to oxygen at one end of the molecule. A hydroxyl OH group is attached to each of the other residues. In glucose, the hydroxyl group attached to the third carbon is on the left side of the molecular structure, and all other hydroxyl groups are on the right. In galactose, the hydroxyl groups attached to the third and fourth carbons are on the left, and the hydroxyl groups attached to the second, fifth, and sixth carbon are on the right. Fructose is a ketose with C doubled bonded to O at the second carbon. All other carbons have hydroxyl groups associated with them. The hydroxyl group associated with the third carbon is on the left, and all the other hydroxyl groups are on the right. Figure 3.5 glucose, galactose, and fructose are all hexoses. They are structural isomers, meaning they have the same chemical formula, C6H12O6, but a different atom arrangement. What kind of sugars are these, aldose or ketose? Glucose, galactose, and fructose are isomeric monosaccharides, hexoses, meaning they have the same chemical formula but have slightly different structures. Glucose and galactose are aldoses, and fructose is a ketose. Monosaccharides can exist as a linear chain or as ring-shaped molecules. In aqueous solutions, they are usually in ring forms, figure 3.6. Glucose in a ring form can have two different hydroxyl group arrangements. OH, around the anomeric carbon, carbon 1, that becomes asymmetric in the ring formation process. If the hydroxyl group is below carbon number 1 in the sugar, it is in the alpha-alpha position, and if it is above the plane, it is in the beta-beta position. The conversion of glucose between linear and ring forms is shown. The glucose ring has 5 carbons and an oxygen. In alpha-glucose, the first hydroxyl group is locked in a down position, and in beta-glucose, the ring is locked in an up position. Structures for ring forms of ribose and fructose are also shown. Both sugars have a ring with 4 carbons and an oxygen. Figure 3.6.5 and 6 carbon monosaccharides exist in equilibrium between linear and ring forms. When the ring forms, the side chain it closes on locks into an alpha or beta position. Fructose and ribose also form rings, although they form 5-membered rings as opposed to the 6-membered ring of glucose. Disaccharides. Disaccharides, D equals 2, form when 2 monosaccharides undergo a dehydration reaction, or a condensation reaction or dehydration synthesis. During this process, one monosaccharide's hydroxyl group combines with another monosaccharide's hydrogen, releasing a water molecule and forming a covalent bond. A covalent bond forms between a carbohydrate molecule and another molecule, in this case, between 2 monosaccharides. Scientists call this a glycosidic bond, figure 3.7. Glycosidic bonds, or glycosidic linkages, can be an alpha or beta type. An alpha bond is formed when the OH group on the carbon 1 of the first glucose is below the ring plane, and a beta bond is formed when the OH group on the carbon 1 is above the ring plane. The formation of sucrose from glucose and fructose is shown. In sucrose, the number 1 carbon of the glucose ring is connected to the number 2 carbon of fructose via an oxygen. Figure 3. 7 sucrose forms when a glucose monomer and a fructose monomer join in a dehydration reaction to form a glycosidic bond. In the process, a water molecule is lost. By convention, the carbon atoms in a monosaccharide are numbered from the terminal carbon closest to the carbonyl group. In sucrose, a glycosidic linkage forms between carbon 1 in glucose and carbon 2 in fructose. Common disaccharides include lactose, maltose, and sucrose, figure 3. 8. Lactose is a disaccharide consisting of the monomers glucose and galactose. It is naturally in milk. Maltose, or malt sugar, is a disaccharide formed by a dehydration reaction between 2 glucose molecules. The most common disaccharide is sucrose, or table sugar, which is comprised of glucose and fructose monomers. The chemical structures of maltose, lactose, and sucrose are shown. Both maltose and lactose are made from 2 glucose monomers joined together in ring form. In maltose, the oxygen in the glycosidic bond points downward. In lactose, the oxygen in the glycosidic bond points upward. Sucrose is made from glucose and fructose monomers. The oxygen in the glycosidic bond points downward. Figure 3. 8 common disaccharides include maltose, grain sugar, lactose, milk sugar, and sucrose, table sugar. Polysaccharides. A long chain of monosaccharides linked by glycosidic bonds is a polysaccharide, poly. Many. The chain may be branched or unbranched, and it may contain different types of monosaccharides. The molecular weight may be 100,000 daltons or more depending on the number of joined monomers. Starch, glycogen, cellulose, and chitin are primary examples of polysaccharides. Plants store sugars in the form of starch. In plants, an amylose and amylopectin mixture, both glucose polymers, comprise these sugars. Plants are able to synthesize glucose, and they store the excess glucose beyond their immediate energy needs as starch in different plant parts, including roots and seeds. The starch in the seeds provides food for the embryo as it germinates and can also act as a food source for humans and animals. Enzymes break down the starch that humans consume. For example, an amylase present in saliva catalyzes or breaks down this starch into smaller molecules, such as maltose and glucose. The cells can then absorb the glucose. Glucose starch comprises monomers that are joined by alpha-1,4 or alpha-1,6 glycosidic bonds. The numbers 1,4 and 1,6 refer to the carbon number of the two residues that have joined to form the bond. As Figure 3.9 illustrates, unbranched glucose monomer chains, only alpha-1,4 linkages, form the starch. Whereas amylopectin is a branched polysaccharide, alpha-1,6 linkages at the branch points. The chemical structures of amylose and amylopectin are shown. Amylose consists of unbranched chains of glucose subunits, and amylopectin consists of branched chains of glucose subunits. Figure 3.9 amylose and amylopectin are two different starch forms. Unbranched glucose monomer chains comprise amylose by alpha-1,4 glycosidic linkages. Branched glucose monomer chains comprise amylopectin by alpha-1,4 and alpha-1,6 glycosidic linkages. Because of the way the subunits are joined, the glucose chains have a helical structure. Glycogen, not shown, is similar in structure to amylopectin but more highly branched. Glycogen is the storage form of glucose in humans and other vertebrates and is comprised of monomers of glucose. Glycogen is the animal equivalent of starch and is a highly branched molecule usually stored in liver and muscle cells. Whenever blood glucose levels decrease, glycogen breaks down to release glucose in a process scientists call glycogenolysis. Cellulose is the most abundant natural biopolymer. Cellulose mostly comprises a plant's cell wall. This provides the cell structural support. Wood and paper are mostly cellulosic in nature. Glucose monomers comprise cellulose that beta-1,4 glycosidic bonds link, Figure 3.10. Three different magnifications of cellulose are shown. The microfibril is shown as a string-like tube. Further magnification shows that the microfibril is made up of chains of glucose molecules. Further magnification shows the structure of the glucose and that they are linked by hydrogen bonds. Figure 3.10 cellulose is an organic compound composed of linear chains of hundreds to thousands of linked glucose molecules. The glucose monomers form hydrogen bonds, holding the chains firmly together side by side and form strong microfibrils. This rigidity is an important structural component of the cell walls found in plants. Credit: Ryan, K. Rao, A. and Hawkins, A. Department of Biology, Texas A&M University. As Figure 3.10 shows, every other glucose monomer in cellulose is flipped over, and the monomers are packed tightly as extended long chains. This gives cellulose its rigidity and high tensile strength, which is so important to plant cells. While human digestive enzymes cannot break down the beta-1,4 linkage, herbivores such as cows, koalas, and buffaloes are able, with the help of the specialized flora in their stomach, to digest plant material that is rich in cellulose and use it as a food source. In some of these animals, certain species of bacteria and protists reside in the rumen part of the herbivore's digestive system and secrete the enzyme cellulase. The appendix of grazing animals also contains bacteria that digest cellulose, giving it an important role in ruminants' digestive systems. Cellulases can break down cellulose into glucose monomers that animals use as an energy source. Termites are also able to break down cellulose because of the presence of other organisms in their bodies that secrete cellulases. Carbohydrates serve various functions in different animals. Arthropods, insects, crustaceans, and others have an outer skeleton, the exoskeleton, which protects their internal body parts, as we see in the bee in Figure 3.11. This exoskeleton is made of the biological macromolecule chitin, which is a nitrogen-containing polysaccharide. It is made of repeating N-acetyl beta-D-glucosamine units, which are a modified sugar. Chitin is also a major component of fungal cell walls. Fungi are neither animals nor plants and form a kingdom of their own in the domain Eukarya. A photograph shows a bee in flight, getting nectar from a flower. Figure 3. 11 insects have a hard outer exoskeleton made of chitin, a type of polysaccharide. Credit, Louise Docker. Career connection. Registered dietitian. Nutrition is a worldwide health concern, and many diseases, such as diabetes and heart disease, are becoming more prevalent. This is one of the reasons why people increasingly seek out registered dietitians for advice. Registered dietitians help plan nutrition programs for individuals in various settings. They often work with patients in healthcare facilities, designing nutrition plans to treat and prevent diseases. For example, dietitians may teach a patient with diabetes how to manage blood sugar levels by eating the correct types and amounts of carbohydrates. Dietitians may also work in nursing homes, schools, and private practices. To become a registered dietitian, one needs to earn at least a bachelor's degree in dietetics, nutrition, food technology, or a related field. In addition, registered dietitians must complete a supervised internship program and pass a national exam. Those who pursue careers in dietetics take courses in nutrition, chemistry, biochemistry, biology, microbiology, and human physiology. Dietitians must become experts in the chemistry and physiology, biological functions, of food, proteins, carbohydrates, and fats. Benefits of carbohydrates. Are carbohydrates good for you? Some people believe that carbohydrates are bad and they should avoid them. Some diets completely forbid carbohydrate consumption, claiming that a low-carbohydrate diet helps people to lose weight faster. However, carbohydrates have been an important part of the human diet for thousands of years. Artifacts from ancient civilizations show the presence of wheat, rice, and corn in our ancestors' storage areas. As part of a well-balanced diet, we should supplement carbohydrates with proteins, vitamins, and fats. Calorie-wise, a gram of carbohydrate provides 4.3 kcal. For comparison, fats provide 9 kcal/g, a less desirable ratio. Carbohydrates contain soluble and insoluble elements. The insoluble part, fiber, is mostly cellulose. Fiber has many uses. It promotes regular bowel movement by adding bulk, and it regulates the blood glucose consumption rate. Fiber also helps to remove excess cholesterol from the body. Fiber binds to the cholesterol in the small intestine, then attaches to the cholesterol and prevents the cholesterol particles from entering the bloodstream. Cholesterol then exits the body via the feces. Fiber-rich diets also have a protective role in reducing the occurrence of colon cancer. In addition, a meal containing whole grains and vegetables gives a feeling of fullness. As an immediate source of energy, glucose breaks down during the cellular respiration process, which produces ATP, the cell's energy currency. Without consuming carbohydrates, we reduce the availability of instant energy. Eliminating carbohydrates from the diet may be necessary for some people, but such a step may not be healthy for everyone. Link to learning. For an additional perspective on carbohydrates, explore biomolecules, the carbohydrates, through this interactive animation. Learning objectives. By the end of this section, you will be able to do the following. Describe the 4 major types of lipids. Explain the role of fats in storing energy. Differentiate between saturated and unsaturated fatty acids. Describe phospholipids and their role in cells. Define the basic structure of a steroid and some steroid functions. Explain how cholesterol helps maintain the plasma membrane's fluid nature. Lipids include a diverse group of compounds that are largely nonpolar in nature. This is because they are hydrocarbons that include mostly nonpolar carbon or carbon-hydrogen bonds. Nonpolar molecules are hydrophobic, water-fearing, or insoluble in water. Lipids perform many different functions in a cell. Cells store energy for long-term use in the form of fats. Lipids also provide insulation from the environment for plants and animals, figure 312. For example, they help keep aquatic birds and mammals dry when forming a protective layer over fur or feathers because of their water-repellent hydrophobic nature. Lipids are also the building blocks of many hormones and are an important constituent of all cellular membranes. Lipids include fats, oils, waxes, phospholipids, and steroids. Photo shows a river otter swimming. The river otter has thick fur that is smooth and wet. Figure 3:12 hydrophobic lipids in aquatic mammals' fur, such as this river otter, protect them from the elements. Credit Ken Bosma. Fats and oils. A fat molecule consists of two main components, glycerol and fatty acids. Glycerol is an organic compound alcohol with 3 carbons, 5 hydrogens, and 3 hydroxyl OH groups. Fatty acids have a long chain of hydrocarbons to which a carboxyl group is attached, hence the name fatty acid. The number of carbons in the fatty acid may range from 4 to 36. The most common are those containing 12-18 carbons. In a fat molecule, the fatty acids attach to each of the glycerol molecule's 3 carbons with an ester bond through an oxygen atom. Figure 313. The structures of glycerol, a fatty acid, and a triacylglycerol are shown. Glycerol is a chain of 3 carbons, with a hydroxyl O H group attached to each carbon. A fatty acid has an acetyl C O H group attached to a long carbon chain. In triacylglycerol, a fatty acid is attached to each of glycerol's 3 hydroxyl groups via the carboxyl group. A water molecule is lost in the reaction, so the structure of the linkage is C-O-C, with an oxygen double bonded to the second carbon. Figure 3:13 joining 3 fatty acids to a glycerol backbone in a dehydration reaction forms triacylglycerol. 3 water molecules release in the process. During this ester bond formation, 3 water molecules are released. The 3 fatty acids in the triacylglycerol may be similar or dissimilar. We also call fats triacylglycerols or triglycerides because of their chemical structure. Some fatty acids have common names that specify their origin. For example, palmitic acid, a saturated fatty acid, is derived from the palm tree. Arachidic acid is derived from Arachis hypogea, the scientific name for groundnuts or peanuts. Fatty acids may be saturated or unsaturated. In a fatty acid chain, if there are only single bonds between neighboring carbons in the hydrocarbon chain, the fatty acid is saturated. Saturated fatty acids are saturated with hydrogen. In other words, the number of hydrogen atoms attached to the carbon skeleton is maximized. Stearic acid is an example of a saturated fatty acid, figure 314. The structure of stearic acid is shown. This fatty acid has a hydrocarbon chain 17 residues long attached to an acetyl group. All bonds between the carbons are single bonds. Figure 3:14 stearic acid is a common saturated fatty acid. When the hydrocarbon chain contains a double bond, the fatty acid is unsaturated. Oleic acid is an example of an unsaturated fatty acid, figure 315. The structure of oleic acid is shown. This fatty acid has a hydrocarbon chain 17 residues long attached to an acetyl group. The bond between carbon 8 and carbon 9 is a double bond. Figure 3:15 oleic acid is a common unsaturated fatty acid. Most unsaturated fats are liquid at room temperature. We call these oils. If there is one double bond in the molecule, then it is a monounsaturated fat, e.g. olive oil, and if there is more than one double bond, then it is a polyunsaturated fat, e.g. canola oil. When a fatty acid has no double bonds, it is a saturated fatty acid because it is not possible to add more hydrogen to the chain's carbon atoms. A fat may contain similar or different fatty acids attached to glycerol. Long straight fatty acids with single bonds generally pack tightly and are solid at room temperature. Animal fats with stearic acid and palmitic acid, common in meat, and the fat with butyric acid, common in butter, are examples of saturated fats. Mammals store fats in specialized cells, or adipocytes, where fat globules occupy most of the cell's volume. Plants store fat or oil in many seeds and use them as a source of energy during seedling development. Unsaturated fats or oils are usually of plant origin and contain cis-unsaturated fatty acids. Cis and trans indicate the configuration of the molecule around the double bond. If hydrogens are present in the same plane, it is a cis fat. If the hydrogen atoms are on 2 different planes, It is a trans fat. The cis double bond causes a bend or a kink that prevents the fatty acids from packing tightly, keeping them liquid at room temperature, figure 316. Olive oil, corn oil, canola oil, and cod liver oil are examples of unsaturated fats. Unsaturated fats help to lower blood cholesterol levels, whereas saturated fats might contribute to plaque formation in the arteries. A comparison of saturated and unsaturated fatty acids is shown. Stearic acid, a saturated fatty acid, has a hydrocarbon chain 17 residues long attached to an acetyl group. Oleic acid also has a 17 residue hydrocarbon chain, but a double bond exists between the 8th and 9th carbon in the chain. In cis-oleic acid, the hydrogens are on the same side of the double bond. In the cis-oleic acid, the two hydrogens on the same side cause the chain to bend. In trans-oleic acid, they are on opposite sides. Figure 3. 16 saturated fatty acids have hydrocarbon chains connected by single bonds only. Unsaturated fatty acids have one or more double bonds. Each double bond may be in a cis or trans configuration. In the cis configuration, both hydrogens are on the same side of the hydrocarbon chain. In the trans configuration, the hydrogens are on opposite sides. A cis double bond causes a kink in the chain. Trans fats. The food industry artificially hydrogenates oils to make them semi-solid and of a consistency desirable for many processed food products. Simply speaking, hydrogen gas is bubbled through oils to solidify them. During this hydrogenation process, double bonds of the cis conformation in the hydrocarbon chain may convert to double bonds in the trans conformation. Margarine, some types of peanut butter, and shortening are examples of artificially hydrogenated trans fats. Recent studies have shown that an increase in trans fats in the human diet may lead to higher levels of low-density lipoproteins, LDL, or bad cholesterol, which in turn may lead to plaque deposition in the arteries, resulting in heart disease. Many fast food restaurants have recently banned using trans fats, and food labels are required to display the trans fat content. Omega fatty acids. Essential fatty acids are those that the human body requires, but does not synthesize. Consequently, they have to be supplemented through ingestion via the diet. Omega-3 fatty acids, like those in figure 317, fall into this category and are one of only two known for humans. The other is omega-6 fatty acid. These are polyunsaturated fatty acids and are omega-3 because a double bond connects the third carbon from the hydrocarbon chain's end to its neighboring carbon. The molecular structures of alpha-linolenic acid, an omega-3 fatty acid, is shown. Alpha-linolenic acid has 3 double bonds located 8, 11, and 14 residues from the acetyl group. It has a hooked shape. Figure 3. 17 alpha-linolenic acid is an example of an omega-3 fatty acid. It has 3 cis double bonds and, as a result, a curved shape. For clarity, the diagram does not show the carbons. Each singly bonded carbon has 2 hydrogens associated with it, which the diagram also does not show. The farthest carbon away from the carboxyl group is numbered as the omega carbon, and if the double bond is between the third and fourth carbon from that end, it is an omega-3 fatty acid. Nutritionally important because the body does not make them, omega-3 fatty acids include alpha-linolic acid, ALA, icosapentenoic acid, EPA, and docosahexaenoic acid, DHA, all of which are polyunsaturated. Salmon, trout, and tuna are good sources of omega-3 fatty acids. Research indicates that omega-3 fatty acids reduce the risk of sudden death from heart attacks, lower triglycerides in the blood, decrease blood pressure, and prevent thrombosis by inhibiting blood clotting. They also reduce inflammation and may help lower the risk of some cancers in animals. Like carbohydrates, fats have received considerable bad publicity. It is true that eating in excess of fried foods and other fatty foods leads to weight gain. However, fats do have important functions. Many vitamins are fat-soluble, and fats serve as a long-term storage form of fatty acids, a source of energy. They also provide insulation for the body. Therefore, we should consume healthy fats in moderate amounts on a regular basis. Waxes. Wax covers some aquatic birds' feathers and some plants' leaf surfaces. Because of wax's hydrophobic nature, they prevent water from sticking on the surface, figure 3. 18. Long fatty acid chains esterified to long-chain alcohols comprise waxes. The photo shows leaves on a plant. The leaves appear thick, shiny, and waxy. Figure 3. 18 lipids comprise waxy coverings on some leaves. Credit: Roger Griffiths. Phospholipids. Phospholipids are major plasma membrane constituents that comprise cells' outermost layer. Like fats, they are comprised of fatty acid chains attached to a glycerol or sphingosine backbone. However, instead of 3 fatty acids attached as in triglycerides, there are 2 fatty acids forming diacylglycerol, and a modified phosphate group occupies the glycerol backbone's 3rd carbon, figure 319. A phosphate group alone attached to a diacylglycerol does not qualify as a phospholipid. It is phosphatidate diacylglycerol 3-phosphate, the precursor of phospholipids. An alcohol modifies the phosphate group. Phosphatidylcholine and phosphatidylserine are 2 important phospholipids that are in plasma membranes. The molecular structure of a phospholipid is shown. It consists of 2 fatty acids attached to the 1st and 2nd carbons in glycerol, and a phosphate group attached to the 3rd position. In the illustration, one of the fatty acids is saturated and straight, and the second is unsaturated and bent. Both are below the glycerol molecule. The phosphate group may be further modified by addition of another molecule to one of its oxygens indicated by uppercase R. 2 molecules may modify the phosphate group, choline and serine. The glycerol and phosphate group make up the hydrophilic head of the molecule, while the fatty acids attached below make up the hydrophobic tails. Figure 3. 19. A phospholipid is a molecule with 2 fatty acids and a modified phosphate group attached to a glycerol backbone. Adding a charged or polar chemical group may modify the phosphate. A phospholipid is an amphipathic molecule, meaning it has a hydrophobic and a hydrophilic part. The fatty acid chains are hydrophobic and cannot interact with water, whereas the phosphate-containing group is hydrophilic and interacts with water, figure 320. An illustration of a phospholipids bilayer is shown. The phospholipids bilayer consists of 2 layers of phospholipids. The hydrophobic tails of the phospholipids face one another, while the hydrophilic head groups face outward. Figure 320, the phospholipid bilayer is the major component of all cellular membranes. The hydrophilic head groups of the phospholipids face the aqueous solution. The hydrophobic tails are sequestered in the middle of the bilayer. The head is the hydrophilic part, and the tail contains the hydrophobic fatty acids. In a membrane, a bilayer of phospholipids forms the structure's matrix. Phospholipids' fatty acid tails face inside, away from water, whereas the phosphate group faces the outside, aqueous side, figure 320. Phospholipids are responsible for the plasma membrane's dynamic nature. If a drop of phospholipids is placed in water, it spontaneously forms a structure that scientists call a micelle, where the hydrophilic phosphate heads face the outside and the fatty acids face the structure's interior. Steroids. Unlike the phospholipids and fats that we discussed earlier, steroids have a fused ring structure. Although they do not resemble the other lipids, scientists group them with them because they are also hydrophobic and insoluble in water. All steroids have 4 linked carbon rings, and several of them, like cholesterol, have a short tail, figure 321. Many steroids also have the O functional group, which puts them in the alcohol classification, sterols. The structures of cholesterol and cortisol are shown. Each of these molecules is composed of 3 6-carbon rings fused to a 5-carbon ring. Cholesterol has a branched hydrocarbon attached to the 5-carbon ring and a hydroxyl group attached to the terminal 6-carbon ring. Cortisol has a 2-carbon chain modified with a double-bonded oxygen, a hydroxyl group attached to the 5-carbon ring, and an oxygen double-bonded to the terminal 6-carbon ring. Figure 3. 21-4 fused hydrocarbon rings comprise steroids such as cholesterol and cortisol. Cholesterol is the most common steroid. The liver synthesizes cholesterol and is the precursor to many steroid hormones such as testosterone and estradiol, which gonads and endocrine glands secrete. It is also the precursor to vitamin D. Cholesterol is also the precursor of bile salts, which help emulsifying fats and their subsequent absorption by cells. Although laypeople often speak negatively about cholesterol, it is necessary for the body's proper functioning. Sterols, cholesterol in animal cells, phytosterol in plants, are components of the plasma membrane of cells and are found within the phospholipid bilayer. Link to learning. For an additional perspective on lipids, watch this video about types of fat. Learning objectives. By the end of this section, you will be able to do the following. Describe the functions proteins perform in the cell and in tissues. Discuss the relationship between amino acids and proteins. Explain the 4 levels of protein organization. Describe the ways in which protein shape and function are linked. Proteins are one of the most abundant organic molecules in living systems and have the most diverse range of functions of all macromolecules. Proteins may be structural, regulatory, contractile, or protective. They may serve in transport, storage, or membranes, or they may be toxins or enzymes. Each cell in a living system may contain thousands of proteins, each with a unique function. Their structures, like their functions, vary greatly. They are all, however, amino acid polymers arranged in a linear sequence. Types and functions of proteins. Enzymes, which living cells produce, are catalysts in biochemical reactions, like digestion, and are usually complex or conjugated proteins. Each enzyme is specific for the substrate, a reactant that binds to an enzyme upon which it acts. The enzyme may help in breakdown, rearrangement, or synthesis reactions. We call enzymes that break down their substrates catabolic enzymes. Those that build more complex molecules from their substrates are anabolic enzymes, and enzymes that affect the rate of reaction are catalytic enzymes. Note that all enzymes increase the reaction rate and therefore are organic catalysts. An example of an enzyme is salivary amylase, which hydrolyzes its substrate amylose, a component of starch. Hormones are chemical signaling molecules, usually small proteins or steroids, secreted by endocrine cells that act to control or regulate specific physiological processes, including growth, development, metabolism, and reproduction. For example, insulin is a protein hormone that helps regulate the blood glucose level. Table 3.1 lists the primary types and functions of proteins. Protein types and functions. Type examples functions. Digestive enzymes amylase, lipase, pepsin, trypsin help in food by catabolizing nutrients into monomeric units. Transport hemoglobin. Albumin carries substances in the blood or lymph throughout the body. Structural actin, tubulin, keratin construct different structures, like the cytoskeleton. Hormones insulin, thyroxine coordinate different body systems activity. Defense immunoglobulins protect the body from foreign pathogens. Contractile actin, myosin affect muscle contraction. Storage legume storage proteins, egg white albumin, provide nourishment in early embryo development and the seedling. Table 3.1. Proteins have different shapes and molecular weights. Some proteins are globular in shape, whereas others are fibrous in nature. For example, hemoglobin is a globular protein, but collagen, located in our skin, is a fibrous protein. Protein shape is critical to its function, and many different types of chemical bonds maintain this shape. Changes in temperature, pH, and exposure to chemicals may lead to permanent changes in the protein's shape, leading to loss of function or denaturation. Different arrangements of the same 20 types of amino acids comprise all proteins. 2 rare new amino acids were discovered recently, selenoscistine and pyrrolysine, and additional new discoveries may be added to the list. Amino acids. Amino acids are the monomers that comprise proteins. Each amino acid has the same fundamental structure, which consists of a central carbon atom or the alpha-alpha carbon, bonded to an amino group, NH2, a carboxyl group, COOH, and to a hydrogen atom. Every amino acid also has another atom or group of atoms bonded to the central atom, known as the R group. Figure 322. The molecular structure of an amino acid is given. An amino acid has an alpha carbon to which an amino group, a carboxyl group, a hydrogen, and a side chain are attached. The side chain varies for different amino acids and is designated as the R group. Figure 3. 22 amino acids have a central asymmetric carbon to which an amino group, a carboxyl group, a hydrogen atom, and a side chain R group are attached. Scientists use the name amino acid because these acids contain both amino group and carboxyl acid group in their basic structure. As we mentioned, there are 20 common amino acids present in proteins. 9 of these are essential amino acids in humans because the human body cannot produce them, and we obtain them from our diet. For each amino acid, the R group, or side chain, is different, figure 323. Visual connection. The molecular structures of the 20 amino acids commonly found in proteins are given. These are divided into 5 categories: nonpolar aliphatic, polar uncharged, positively charged, negatively charged, and aromatic. Nonpolar aliphatic amino acids include glycine, alanine, valine, leucine, methionine, isoleucine, and proline. Polar uncharged amino acids include serine, threonine, cysteine, asparagine, and glutamine. Positively charged amino acids include lysine, arginine, and histidine. Negatively charged amino acids include aspartate and glutamate. Aromatic amino acids include phenylalanine, tyrosine, and tryptophan. For example, in the amino acid glycine, the R group is a single hydrogen, but in alanine, the R group is upper C upper H subscript 3 baseline. Figure 3.23. There are 20 common amino acids commonly found in proteins, each with a different R group, variant group, that determines its chemical nature. Which categories of amino acid would you expect to find on a soluble protein surface, and which would you expect to find in the interior? What distribution of amino acids would you expect to find in a protein embedded in a lipid bilayer? The chemical nature of the side chain determines the amino acid's nature, that is, whether it is acidic, basic, polar, or nonpolar. For example, the amino acid glycine has a hydrogen atom as the R group. Amino acids such as valine, methionine, and alanine are nonpolar or hydrophobic in nature, while amino acids such as serine, threonine, and cysteine are polar and have hydrophilic side chains. The side chains of lysine and arginine are positively charged, and therefore these amino acids are also basic amino acids. Proline has an R group that is linked to the amino group, forming a ring-like structure. Proline is an exception to the amino acid standard structure, since its amino group is not separate from the side chain, figure 323. A single uppercase letter or a 3-letter abbreviation represents amino acids. For example, the letter V or the 3-letter symbol VAL represent valine. Just as some fatty acids are essential to a diet, some amino acids also are necessary. These essential amino acids in humans include isoleucine, leucine, and cysteine. Essential amino acids refer to those necessary to build proteins in the body, but not those that the body produces. Which amino acids are essential varies from organism to organism. The sequence and the number of amino acids ultimately determine the protein's shape, size, and function. A covalent bond, or peptide bond, attaches to each amino acid, which a dehydration reaction forms. One amino acid's carboxyl group and the incoming amino acid's amino group combine, releasing a water molecule. The resulting bond is the peptide bond, figure 324. The formation of a peptide bond between 2 amino acids is shown. When the peptide bond forms, the carbon from the carbonyl group becomes attached to the nitrogen from the amino group. The upper O, upper H from the carboxyl group and an upper H from the amino group form a molecule of water. Figure 324 peptide bond formation is a dehydration synthesis reaction. The carboxyl group of one amino acid is linked to the incoming amino acid's amino group. In the process, it releases a water molecule. The products that such linkages form are peptides. As more amino acids join to this growing chain, the resulting chain is a polypeptide. Each polypeptide has a free amino group at one end. This end is called the N-terminal or the amino terminal, and the other end has a free carboxyl group, also called the C or carboxyl terminal. While the terms polypeptide and protein are sometimes used interchangeably, a polypeptide is technically a polymer of amino acids, whereas the term protein is used for a polypeptide or polypeptides that have combined together, often have bound non-peptide prosthetic groups, have a distinct shape, and have a unique function. After protein synthesis, translation, most proteins are modified. These are known as post-translational modifications. They may undergo cleavage, phosphorylation, or may require adding other chemical groups. Only after these modifications is the protein completely functional. Link to learning. Click through the steps of protein synthesis in this interactive tutorial. Evolution connection. The evolutionary significance of cytochrome C. Cytochrome C is an important component of the electron transport chain, a part of cellular respiration, and it is normally located in the cellular organelle, the mitochondrion. This protein has a heme prosthetic group, and the heme's central ion alternately reduces and oxidizes during electron transfer. Because this essential protein's role in producing cellular energy is crucial, it has changed very little over millions of years. Protein sequencing has shown that there is a considerable amount of cytochrome C amino acid sequence homology among different species. In other words, we can assess evolutionary kinship by measuring the similarities or differences among various species, DNA, or protein sequences. Scientists have determined that human cytochrome C contains 104 amino acids. For each cytochrome C molecule from different organisms that scientists have sequenced to date, 37 of these amino acids appear in the same position in all cytochrome C samples. This indicates that there may have been a common ancestor. On comparing the human and chimpanzee protein sequences, scientists did not find a sequence difference. When researchers compared human and rhesus monkey sequences, the single difference was in one amino acid. In another comparison, human to yeast sequencing shows a difference in the 44th position. Protein structure. As we discussed earlier, a protein's shape is critical to its function. For example, an enzyme can bind to a specific substrate at an active site. If this active site is altered because of local changes or changes in overall protein structure, the enzyme may be unable to bind to the substrate. To understand how the protein gets its final shape or conformation, we need to understand the 4 levels of protein structure: primary, secondary, tertiary, and quaternary. Primary structure. Amino acid's unique sequence in a polypeptide chain is its primary structure. For example, the pancreatic hormone insulin has 2 polypeptide chains, A and B, and they are linked together by disulfide bonds. The N-terminal amino acid of the chain is glycine, whereas the C-terminal amino acid is asparagine, figure 325. The amino acid sequences in the A and B chains are unique to insulin. The amino acid sequences for the chain and B chain of bovine insulin are shown. The chain is 21 amino acids in length, and the B chain is 30 amino acids in length. One disulfide, or SS bond, connects 2 cysteine residues in the chain. 2 other disulfide linkages connect the chain to the B chain. Figure 325. Bovine serum insulin is a protein hormone comprised of 2 peptide chains, a 21 amino acids long and B 30 amino acids long. In each chain, 3-letter abbreviations that represent the amino acids' names in the order they are present indicate primary structure. The amino acid cysteine, size, has a sulfhydryl SH group as a side chain. 2 sulfhydryl groups can react in the presence of oxygen to form a disulfide SS bond. 2 disulfide bonds connect the A and B chains together, and a third helps the chain fold into the correct shape. Note that all disulfide bonds are the same length, but we have drawn them different sizes for clarity. The gene encoding the protein ultimately determines the unique sequence for every protein. A change in nucleotide sequence of the gene's coding region may lead to adding a different amino acid to the growing polypeptide chain, causing a change in protein structure and function. In sickle cell anemia, the hemoglobin beta chain, a small portion of which we show in figure 326, has a single amino acid substitution, causing a change in protein structure and function. Specifically, valine in the beta chain substitutes the amino acid glutamic. What is most remarkable to consider is that a hemoglobin molecule is comprised of 2 alpha and 2 beta chains that each consist of about 150 amino acids. The molecule, therefore, has about 600 amino acids. The structural difference between a normal hemoglobin molecule and a sickle cell molecule, which dramatically decreases life expectancy, is a single amino acid of the 600. What is even more remarkable is that 3 nucleotides each encode those 600 amino acids, and a single base change, point mutation. 1 in 1800 bases causes the mutation. Several representations of hemoglobin proteins are shown under normal and sickle cell conditions. In primary structure, the 6th amino acid is replaced by valine. In secondary and tertiary structures, the sickle cell is revealed by a misshapen molecule. As a result, in function, normal hemoglobins do not associate with each other, and each can carry oxygen. But in sickle cell conditions, the proteins aggregate into a fiber and oxygen carrying capacity is reduced. Figure 3. 26. Because of this change of one amino acid in the chain, hemoglobin molecules form long fibers that distort the biconcave, or disc-shaped, red blood cells and causes them to assume a crescent or sickle shape, which clogs blood vessels. Figure 3. 27. The beta-beta chain of hemoglobin is 147 amino acids in length, yet a single amino acid substitution in the primary sequence leads to changes in secondary, tertiary, and quaternary structures and sickle cell anemia. In normal hemoglobin, the amino acid at position 6 is glutamate. In sickle cell, hemoglobin glutamate is replaced by valine. Credit: Rao A. Tag A. Ryan K. and Fletcher S. Department of Biology, Texas A&M University. Because of this change of one amino acid in the chain, hemoglobin molecules form long fibers that distort the biconcave, or disc-shaped, red blood cells and causes them to assume a crescent or sickle shape, which clogs blood vessels. Figure 3. 27. This can lead to myriad serious health problems such as breathlessness, dizziness, headaches, and abdominal pain for those affected by this disease. William Warwick Cardozo showed that sickle cell anemia is an inherited disorder, meaning that the difference in the specific genes encoding region is passed down from parents to children. As you will learn in the genetics unit, the inheritance of such traits is determined by a combination of genes from both parents, and these very small differences can have significant impacts on organisms. This electron micrograph shows red blood cells from a patient with sickle cell anemia. Most of the cells have a normal disc shape, but about 1 in 5 has a sickle shape. A normal blood cell is 8 microns across. Figure 3. 27. In this blood smear, visualized at 535x magnification using bright field microscopy, sickle cells are crescent-shaped, while normal cells are disc-shaped. Credit: Modification of work by Ed Uthman. Scale bar data from Matt Russell. Secondary structure: the local folding of the polypeptide in some regions gives rise to the secondary structure of the protein. The most common are the alpha helix and beta pleated sheet structures, figure 328. Both structures are held in shape by hydrogen bonds. The hydrogen bonds form between the oxygen atom in the carbonyl group in one amino acid and another amino acid that is 4 amino acids farther along the chain. The illustration shows an alpha helix protein structure, which coils like a spring, and a beta pleated sheet structure, which forms flat sheets stacked together. In an alpha helix, hydrogen bonding occurs between the carbonyl group of one amino acid and the amino group of the amino acid that occurs 4 residues later. In a beta-pleated sheet, hydrogen bonding occurs between 2 different lengths of peptide that are anti-parallel to one another. Figure 3. 28. The alpha helix and beta-pleated sheet are secondary protein structures formed when hydrogen bonds form between the carbonyl oxygen and the amino hydrogen in the peptide backbone. Certain amino acids have a propensity to form an alpha helix while others favor beta-pleated sheet formation. Black equals carbon, white equals hydrogen, blue equals nitrogen, and red equals oxygen. Credit: Rao A. Ryan K. Fletcher S. and Tag A. Department of Biology, Texas A&M University. Every helical turn in an alpha helix has 3 6-amino acid residues. The polypeptides R groups, the variant groups, protrude out from the alpha helix chain. In the beta-pleated sheet, hydrogen bonding between atoms on the polypeptide chain's backbone form the pleats. The R groups are attached to the carbons and extend above and below the pleats folds. The pleated segments align parallel or anti-parallel to each other, and hydrogen bonds form between the partially positive hydrogen atom in the amino group and the partially negative oxygen atom in the peptide backbone's carbonyl group. The alpha helix and beta-pleated sheet structures are in most globular and fibrous proteins, and they play an important structural role. Tertiary structure. The polypeptide's unique three-dimensional structure is its tertiary structure, figure 3.29. This structure is in part due to chemical interactions at work on the polypeptide chain. Primarily, the interactions among R groups create the protein's complex three-dimensional tertiary structure. The nature of the R groups in the amino acids involved can counteract forming the hydrogen bonds we described for standard secondary structures. For example, R groups with like charges repel each other, and those with unlike charges are attracted to each other, ionic bonds. When protein folding takes place, the nonpolar amino acids' hydrophobic R groups lie in the protein's interior, whereas the hydrophilic R groups lie on the outside. Scientists also call the former interaction types hydrophobic interactions. Interaction between cysteine side chains forms disulfide linkages in the presence of oxygen, the only covalent bond that forms during protein folding. This illustration shows a polypeptide backbone folded into a three-dimensional structure. Chemical interactions between amino acid side chains maintain its shape. These include an ionic bond between an amino group and a carboxyl group, hydrophobic interactions between two hydrophobic side chains, a hydrogen bond between a hydroxyl group and a carbonyl group, and a disulfide linkage. Figure 3.29, a variety of chemical interactions determine the protein's tertiary structure. These include hydrophobic interactions, ionic bonding, hydrogen bonding, and disulfide linkages. All of these interactions, weak and strong, determine the protein's final three-dimensional shape. When a protein loses its three-dimensional shape, it may no longer be functional. Quaternary structure. In nature, some proteins form from several polypeptides, or subunits, and the interaction of these subunits forms the quaternary structure. Weak interactions between the subunits help to stabilize the overall structure. For example, insulin, a globular protein, has a combination of hydrogen and disulfide bonds that cause it to mostly clump into a ball shape. Insulin starts out as a single polypeptide and loses some internal sequences in the presence of post-translational modification after forming the disulfide linkages that hold the remaining chains together. Silk, a fibrous protein, however, has a beta-pleated sheet structure that is the result of hydrogen bonding between different chains. Figure 3.30 illustrates the four levels of protein structure: primary, secondary, tertiary, and quaternary. Shown are the four levels of protein structure. The primary structure is the amino acid sequence. Secondary structure is a regular folding pattern due to hydrogen bonding. Tertiary structure is the three-dimensional folding pattern of the protein due to interactions between amino acid side chains. Quaternary structure is the interaction of two or more polypeptide chains. Figure 3.30 observe the four levels of protein structure in these illustrations. Credit: Rao, A. Ryan, K. and Tag, A. Department of Biology, Texas A&M University. Denaturation and protein folding. Each protein has its own unique sequence and shape that chemical interactions hold together. If the protein is subject to changes in temperature, pH, or exposure to chemicals, the protein structure may change, losing its shape without losing its primary sequence in what scientists call denaturation. Denaturation is often reversible because the polypeptide's primary structure is conserved in the process if the denaturing agent is removed, allowing the protein to resume its function. Sometimes denaturation is irreversible, leading to loss of function. One example of irreversible protein denaturation is frying an egg. The albumin protein in the liquid egg white denatures when placed in a hot pan. Not all proteins denature at high temperatures. For instance, bacteria that survive in hot springs have proteins that function at temperatures close to boiling. The stomach is also very acidic, has a low pH, and denatures proteins as part of the digestion process. However, the stomach's digestive enzymes retain their activity under these conditions. Protein folding is critical to its function. Scientists originally thought that the proteins themselves were responsible for the folding process. Only recently researchers discovered that often they receive assistance in the folding process from protein helpers, or chaperones, or chaperonins, that associate with the target protein during the folding process. They act by preventing polypeptide aggregation that comprise the complete protein structure, and they dissociate from the protein once the target protein is folded. Link to learning. For an additional perspective on proteins, view this animation called Biomolecules, the proteins. Learning objectives. By the end of this section, you will be able to do the following. Describe nucleic acid's structure and define the two types of nucleic acids. Explain DNA's structure and role. Explain RNA's structure and roles. Nucleic acids are the most important macromolecules for the continuity of life. They carry the cell's genetic blueprint and carry instructions for its functioning. DNA and RNA. The two main types of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA is the genetic material in all living organisms, ranging from single-celled bacteria to multicellular mammals. It is in the nucleus of eukaryotes and in the organelles, chloroplasts, and mitochondria. In prokaryotes, the DNA is not enclosed in a membranous envelope. The cell's entire genetic content is its genome, and the study of genomes is genomics. In eukaryotic cells, but not in prokaryotes, DNA forms a complex with histone proteins to form chromatin, the substance of eukaryotic chromosomes. A chromosome may contain tens of thousands of genes. Many genes contain the information to make protein products. Other genes code for RNA products. DNA controls all of the cellular activities by turning the genes on or off. The other type of nucleic acid, RNA, is mostly involved in protein synthesis. The DNA molecules never leave the nucleus, but instead use an intermediary to communicate with the rest of the cell. This intermediary is the messenger RNA, mRNA. Other types of RNA, like rRNA, tRNA, and microRNA, are involved in protein synthesis and its regulation. DNA and RNA are comprised of monomers that scientists call nucleotides. The nucleotides combine with each other to form a polynucleotide, DNA, or RNA. Three components comprise each nucleotide: a nitrogenous base, a pentose, 5-carbon sugar, and a phosphate group, figure 331. Each nitrogenous base in a nucleotide is attached to a sugar molecule, which is attached to one or more phosphate groups. The molecular structure of a nucleotide is shown. The core of the nucleotide is a pentose whose carbon residues are numbered 1' through 5'. The base is attached to the 1' carbon, and the phosphate is attached to the 5' carbon. Two kinds of pentose are found in nucleotides, ribose and deoxyribose. Deoxyribose has an H instead of OH at the 2' position. 5 kinds of base are found in nucleotides. Two of these, adenine and guanine, are purine bases with 2 rings fused together. The other 3, cytosine, thymine, and uracil, have one 6-membered ring. Figure 331. 3 components comprise a nucleotide: a nitrogenous base, a pentose sugar, and one or more phosphate groups. Carbon residues in the pentose are numbered 1 through 5. The prime distinguishes these residues from those in the base, which are numbered without using a prime notation. The base is attached to the ribose's 1 position, and the phosphate is attached to the 5 position. When a polynucleotide forms, the incoming nucleotide's 5-phosphate attaches to the 3-hydroxyl group at the end of the growing chain. Two types of pentose are in nucleotides: deoxyribose, found in DNA, and ribose, found in RNA. Deoxyribose is similar in structure to ribose, but it has an H instead of an OH at the 2 position. We can divide bases into 2 categories: purines and pyrimidines. Purines have a double ring structure, and pyrimidines have a single ring. The nitrogenous bases, important components of nucleotides, are organic molecules and are so named because they contain carbon and nitrogen. They are bases because they contain an amino group that has the potential of binding an extra hydrogen, and thus decreasing the hydrogen ion concentration in its environment, making it more basic. Each nucleotide in DNA contains one of 4 possible nitrogenous bases: adenine A, guanine G, cytosine C, and thymine T. Scientists classify adenine and guanine as purines. The purine's primary structure is two carbon-nitrogen rings. Scientists classify cytosine, thymine, and uracil as pyrimidines, which have a single carbon-nitrogen ring as their primary structure. Figure 331. Each of these basic carbon-nitrogen rings has different functional groups attached to it. In molecular biology shorthand, we know the nitrogenous bases by their symbols A, T, G, C, and U. DNA contains A, T, G, and C, whereas RNA contains a U, G, and C. The pentose sugar in DNA is deoxyribose, and in RNA the sugar is ribose. Figure 331. The difference between the sugars is the presence of the hydroxyl group on the ribose's second carbon and hydrogen on the deoxyribose's second carbon. The carbon atoms of the sugar molecule are numbered as 1, 2, 3, 4, and 5. 1 is read as 1'. The phosphate residue attaches to the hydroxyl group of the 5-carbon of one sugar and the hydroxyl group of the 3-carbon of the sugar of the next nucleotide, which forms a 5,3 phosphodiester linkage. A simple dehydration reaction, like the other linkages connecting monomers in macromolecules, does not form the phosphodiester linkage. Its formation involves removing two phosphate groups. A polynucleotide may have thousands of such phosphodiester linkages. DNA double helix structure. DNA has a double helix structure. Figure 332. The sugar and phosphate lie on the outside of the helix, forming the DNA's backbone. The nitrogenous bases are stacked in the interior, like a pair of staircase steps. Hydrogen bonds bind the pairs to each other. Every base pair in the double helix is separated from the next base pair by 0.34 nm. The helix's two strands run in opposite directions, meaning that the 5-carbon end of one strand will face the 3-carbon end of its matching strand. Scientists call this an antiparallel orientation and is important to DNA replication and in many nucleic acid interactions. The molecular structure of DNA is shown. DNA consists of two antiparallel strands twisted in a double helix. The phosphate backbone is on the outside, and the nitrogenous bases face one another on the inside. The bases appear as strands between the phosphate backbone of the double helix. Figure 332, native DNA, is an antiparallel double helix. The phosphate backbone, indicated by the curvy lines, is on the outside, and the bases are on the inside. Each base from one strand interacts via hydrogen bonding with a base from the opposing strand. Credit: Jerome Walker Dennis Mitz. Only certain types of base pairing are allowed. For example, a certain purine can only pair with a certain pyrimidine. This means it can pair with T and G can pair with C, as figure 3.33 shows. This is the base complementary rule. In other words, the DNA strands are complementary to each other. If the sequence of one strand is AATTGGCC, the complementary strand would have the sequence TTAACCGG. During DNA replication, each strand copies itself, resulting in a daughter DNA double helix containing one parental DNA strand and a newly synthesized strand. Visual connection: hydrogen bonding between thymine and adenine and between guanine and cytosine is shown. Thymine forms 2 hydrogen bonds with adenine, and guanine forms 3 hydrogen bonds with cytosine. The phosphate backbones of each strand are on the outside and run in opposite directions. Figure 3.33. In a double-stranded DNA molecule, the two strands run anti-parallel to one another so that one strand runs 5 to 3 and the other 3 to 5. The phosphate backbone is located on the outside, and the bases are in the middle. Adenine forms hydrogen bonds, or base pairs, with thymine, and guanine base pairs with cytosine. A mutation occurs, and adenine replaces cytosine. What impact do you think this will have on the DNA structure? RNA. Ribonucleic acid, or RNA, is mainly involved in the process of protein synthesis under the direction of DNA. RNA is usually single-stranded and is comprised of ribonucleotides that are linked by phosphodiester bonds. A ribonucleotide in the RNA chain contains ribose, the pentose sugar, one of the 4 nitrogenous bases, A, U, G, and C, and the phosphate group. There are 4 major types of RNA: messenger RNA, mRNA, ribosomal RNA, rRNA, transfer RNA, tRNA, and microRNA, miRNA. The first, mRNA, carries the message from DNA, which controls all of the cellular activities in a cell. If a cell requires synthesizing a certain protein, the gene for this product turns on, and the messenger RNA synthesizes in the nucleus. The RNA base sequence is complementary to the DNA's coding sequence from which it has been copied. However, in RNA, the base T is absent and U is present instead. If the DNA strand has a sequence AATTGCGC, the sequence of the complementary RNA is UUAACGCG. In the cytoplasm, the mRNA interacts with ribosomes and other cellular machinery. Figure 334. An illustration of a ribosome is shown. mRNA sits between the large and small subunits. tRNA molecules bind the ribosome and add amino acids to the growing peptide chain. Figure 334. A ribosome has two parts, a large subunit and a small subunit. The mRNA sits in between the two subunits. A tRNA molecule recognizes a codon on the mRNA, binds to it by complementary base pairing, and adds the correct amino acid to the growing peptide chain. The mRNA is read in sets of 3 bases known as codons. Each codon codes for a single amino acid. In this way, the mRNA is read and the protein product is made. Ribosomal RNA, rRNA, is a major constituent of ribosomes on which the mRNA binds. The rRNA ensures the proper alignment of the mRNA and the ribosomes. The ribosome's rRNA also has an enzymatic activity, peptidyl transferase, and catalyzes peptide bond formation between two aligned amino acids. Transfer RNA, tRNA, is one of the smallest of the 4 types of RNA, usually 70-90 nucleotides long. It carries the correct amino acid to the protein synthesis site. It is the base pairing between the tRNA and mRNA that allows for the correct amino acid to insert itself in the polypeptide chain. MicroRNAs are the smallest RNA molecules, and their role involves regulating gene expression by interfering with the expression of certain mRNA messages. Table 3.2 summarizes DNA and RNA features. DNA and RNA features. DNA-RNA. Function carries genetic information involved in protein synthesis. Location remains in the nucleus, leaves the nucleus. Structure double helix, usually single-stranded. Sugar deoxyribose, pyrimidine cytosine, thymine cytosine, yours. Purines adenine, guanine. Table 3.2. Even though the RNA is single-stranded, most RNA types show extensive intramolecular base pairing between complementary sequences, creating a predictable 3-dimensional structure essential for their function. As you have learned, information flow in an organism takes place from DNA to RNA to protein. DNA dictates the structure of mRNA in a process scientists call transcription, and RNA dictates the protein's structure in a process scientists call translation. This is the central dogma of life, which holds true for all organisms. However, exceptions to the rule occur in connection with viral infections. Link to learning. To learn more about DNA, explore the Howard Hughes Medical Institute biointeractive animations on the topic of DNA.